Alrighty, it is my pleasure to introduce our next speaker, Dr. Mike Tokach. Dr. Tokach received his PhD in swine nutrition from the University of Minnesota and has been on the faculty at Kansas State University since 1991. Dr. Tokach's passions include training students, conducting large-scale research, and being a resource for those interested in improving the profitability of pork producers. As a member of a prolific applied swine nutrition team at K-State, Tokach has published over 340 journal articles, more than 1,000 extension publications, and received more than $19 million in grants and gifts to Kansas State University. Dr. Tokach has advised over 100 master's and PhD students and was named a University Distinguished Professor in 2013. Please join me in welcoming Dr. Mike Tokach.
Thank you. Well, thank you. It's certainly a pleasure for me to be here. Just like Hans said, to be on this kind of a program is quite an honor, and I want to thank John and the organizing committee for inviting me. Put me in a little bit of a difficult spot being right after Hans Stein on the program and one of the last speakers of the conference. But before I get started, I do want to thank my colleagues, the faculty that are listed up here that on a part of our team that certainly they're responsible for all the work that gets done at K-State, and I'll let them answer all the difficult questions today. So as we get started, I do have two topics I'm supposed to cover today. One of them is the branched-chain amino acids, and the second one is about swine nutrition in general. And what I'm going to talk about there specifically is about training of graduate students and spend just a few minutes about how we go about that at K-State and what I think some of the needs are there in the future. So first, in terms of what I'm going to discuss on branched-chain amino acids, is give you a little bit of background. And so for some of you that have really been studying this subject quite a bit, my first about 15, 20 slides are going to be a lot of review for you. Then I'll try to get into some of the recent research, the work that we've done at K-State, but also some of the work that Hans's group has done at University of Illinois, and some of the work that has come out of South Dakota State University and with working with Paul Klein at Christensen Farms and some of the excellent work that they've done. And then finish up with just a brief current recommendations. So first, in terms of the background, and this kind of summarizes up where we kind of came to about a year and a half ago in terms of our level of knowledge about branched-chain amino acids. The first one is that if we have excess leucine, we know that it stimulates the key enzymes that cause breakdown of the other branched-chain amino acids, namely, of course, valine and isoleucine. The excess leucine also reduces uptake of the large neutral amino acids, and the one of particular importance to us, of course, is tryptophan, and it reduces that uptake by the brain because of competition for transport into the brain. The negative effects of high leucine really became a practical reality for us several years ago when we started using blood products in the diet. And that showed up for us in some of the nursery diets. When we got past about 2.5% blood cells in a nursery diet, we started showing up that we were drastically reducing feed intake, and it was because of a deficiency in isoleucine that was occurring. But then as we moved into the corn byproducts, this again came up as a major issue, in that DDGS, high-protein DDGs, and corn gluten meal will result in these diets also that have great excesses in leucine. And there's really less of a concern in those diets if you have enough soybean meal there. If you have enough intact proteins that you don't rely on a lot of the synthetic amino acids, you don't push the pressure down on valine and isoleucine, and it really doesn't show up as a major concern. But the problem does occur, of course, if you bring in larger levels of those other amino acids, which have become more and more cost-effective for us. So bottom line is that excess branched-chain amino acids increases the requirement for valine and isoleucine and for tryptophan in terms of our normal diet formulation. And really, the practical side of it is that what we've really found in terms of both some of the work I'll show you and our group and some of the others is that providing higher levels of some of those other amino acids certainly shows promise for dealing with some of the negative effects of high leucine, but it's not an end-all be-all. And we know that there are certain levels of branched-chain amino acids of leucine that we get to that we can't bring the pig back with bringing in extra levels of valine, tryptophan, and isoleucine. And so first, take a step back on the branched-chain amino acids. Of course, they're similar in their structure, and they share the first rate-limiting step, and first steps, I should say, in catabolism. And then the excess of one of those amino acids increases the catabolism then of all the rest of them. And a very important piece of that is that leucine is the most potent stimulator of the breakdown of all of the branched chain amino acids. And so that's why an excess of leucine is much worse than having an excess of valine or an excess of isoleucine. From a practical standpoint, we very seldom will have an excess of valine or isoleucine. Again, these also interact with the large neutral amino acids, particularly at the brain border to get into the brain, and we know that all these large neutral amino acids play roles in feed intake regulation. I've got it shown up here probably too simply through serotonin, catecholamines, and histamine. The reality is with serotonin, we're not really sure if that's the only way that tryptophan or if that's the main way that tryptophan is really influencing feed intake regulation, especially when we see the increases in feed intake that we get with increasing tryptophan levels in the diet. But to get down to again is just what one of the conventional thinking is that high leucine levels will lower the tryptophan going into the brain, which alters the serotonin activity, and if either through that or through another pathway, reduces feed intake. There is another mode of action, and Enrique Smin was the one that's done a lot of the work for us in this area and helped point this out to me in terms of the literature showing, is that under normal situations, we know that increasing the leucine level in the diet can be positive because it increases protein deposition. And that certainly occurs, and they know it occurs through this mTOR stimulation. But the problem is when you have high leucine levels, and this is shown in a lot of cancer situations as a good example, is that you get this excessive mTOR stimulation. What excessive mTOR stimulation does, it just becomes a very strong inhibitor of feed intake. And so when you have a situation where you have too much leucine in the diet, and I think this is what some of the data is telling us, if we get to a certain level of leucine, that we're starting to drive mTOR in the pig to a certain level, it doesn't matter what we do with the other amino acids, we're not going to bring them back because they've got such a potent reduction in feed intake caused by this mTOR stimulation. And to give just one example of what was shown in the literature, there's a number of different really good studies that have been done, but this is one of my favorite ones that shows the impact of increasing the leucine to lysine ratio and what impact it has. And so they have a 1-to-1 ratio of leucine to lysine going up to 186 to 1, which we can reach quite easily in a high DDGS diet, or certainly we bring in corn gluten meal. And then levels that we don't normally see in a normal diet, but they used with these, and these were all done with synthetic leucine. And what you would think would be adequate levels of valine, isoleucine, and tryptophan in all these diets. And what you can see is that in feed intake side is this progressive reduction in feed intake, and again, over time, the biggest reduction being with that really high level of leucine. And again, I think this really, again, shows where there's something else driving this beyond what's going on with these branched chain amino acids and large neutral amino acids to drive down that consumption. And to show what's going on inside that pig is here you have the branched chain amino acids in the plasma. And of course, we're feeding more leucine. You get more leucine in the plasma, and it's causing the breakdown of the other branched chain amino acids, and so you get less valine and less isoleucine in circulation. And then you look at the ketoacid dehydrogenase enzyme that's breaking down those amino acids, and you see that increases in pretty much every organ that is measured, that has high enough levels of that enzyme to be measured. And the resulting keto acids in circulation, the keto acid from leucine goes up, but because there's less and less amount of valine and isoleucine being circulated, there's less of those keto acids also in circulation over time. And when you get again up to the brain and what's going on in the brain of those pigs, is you look at the level of leucine is going up in the brain, and it's competing for the same transporters, plus you got less plasma levels of valine and isoleucine, and you can see those dropping quite dramatically in the brain, while tryptophan is also going down again because of that competition for getting into the brain. And then you get the reduction in serotonin, and the reduction in serotonin measured out in the plasma because of the increase in leucine. So again, I think a very nice study that shows that overall effect of what's happening inside the pig. So there's really these three modes of action, at least these three modes of action that we think are important in terms of the reduction in feed intake that you see with excess leucine. The first one being that the leucine leads to catabolism of valine and isoleucine, causing a deficiency in both of those amino acids. The second one is that it competes with tryptophan for transport into the blood-brain barrier and reduces the brain tryptophan levels and thus serotonin production. And the third one being is that, as I mentioned, as you get to excess leucine, it causes excessive mTOR stimulation. So one of the very first people that I think that started to summarize up some of the data with the blood cells and the blood meal, particularly late finishing, and to some of these nursery trials that were done to look at the isoleucine requirement at the time was John Tu with Evonik. And I think he did a really nice job of starting to tell us that there's some issues going on here, is that those trials where they had a lower leucine to lysine ratio, the estimate of the requirement was always down here in the 53 to 56%. But when you got over 150% leucine to lysine ratio, again, most of these were by bringing in blood cells to drive down the isoleucine level in the diet to test the ratio. In those diets, the requirement was averaged up here closer to 60%. And he proposed this quadratic relationship, which is an interesting one to me because that also matches up with the quadratic relationship that Enrique found in some of the meta-analysis that we've been able to put together. But I've always looked at this and I've tried to understand, is this really a quadratic relationship, or is this two different points of you're around 55% down here and you're somewhere around 60 to 61% if you got a high level of leucine up there? And I don't think we have enough knowledge to know that for sure off that. And so again, help those of you that don't deal with corn products all the time on a daily basis and looking at these amino acid ratios to why this becomes really important, is look at the leucine to crude protein ratio in the major corn products and in sorghum grain as compared to barley, wheat bran, and soybean meal. So a lot of these things we're talking about today really aren't much of an issue in Europe. They aren't much of an issue if you're dealing with wheat and barley-based diets in Canada, but they're certainly a major issue for us when we talk about DDGS and corn gluten meal or corn as our major ingredients. And you see that leucine to crude protein ratio leads to a higher leucine to lysine ratio. And even within the branched chain amino acids, we have a much higher leucine to valine and leucine to isoleucine ratio than what you have in those other ingredients, or particularly in soybean meal. And then you can look at the leucine to tryptophan ratio and how extreme that gets in some of these diets. And of course, we can correct for a lot of that tryptophan with synthetic tryptophan bringing in those diets, but it's just being aware of just how high the leucine to tryptophan ratio gets in some of these ingredients. And so I brought up Enrique a few times, and I do give him a tremendous amount of credit for the work he did to help us understand this area as part of his PhD project with us. Is he put together an equation after a meta-analysis of different experiments that had wide ranges of leucine to lysine, isoleucine to valine, and tryptophan to lysine ratios. And just briefly, the major components that come into that equation, besides the body weight of the pig and what that impact that has on average daily gain, the leucine level of the diet, that one is interesting when you look at this, is that it's a positive, but the problem is it comes down here when you look at this portion of the equation where it really becomes negative is when the ratio to those other branched chain amino acids to leucine. And that isoleucine to lysine ratio, also that quadratic effect shows up here and shows that you do get some benefit of isoleucine when you start adding a little bit of extra isoleucine into the diet, but then it turns over and you don't get any further benefit as you go to higher and higher isoleucine levels. Valine is a little more linear, even though this shows up on the quadratic side. When you actually put this equation into practice, it becomes more of a linear prediction of what average daily gain will do as you increase the valine level of the diet. And then you also get a linear improvement predicted with increasing tryptophan in the diet because of the bottom side of this ratio with isoleucine. I do think it's interesting to me that it came out, and we debated this quite a bit, about why this is isoleucine to tryptophan down here and not leucine to tryptophan. And it's basically that's what the models told us in terms of what was available in the data sets. But we do think that there is some other interactions going on in some of these trials, and it may have been some of those blood cell trials that were really low in isoleucine that really started driving some of this or of this effect being seen down here. But really the bottom line of this is that it suggests that you can recover some of the lost growth performance with high leucine diets by adding valine, isoleucine, or tryptophan, or multiple combinations of that. And so that's a lot of the background, and I'll walk you through what we've learned since Enrique put together some of that equation and some of this knowledge that we had. Well, the first trial we did was one that Hayden Kirkhart presented at Midwest Animal Science meetings, where he replaced some of that soybean meal with DDGS and synthetic amino acids to get lower amino acid ratios, increase those amino acid ratios a little bit, or added valine, isoleucine, or tryptophan. And the purple is what actually happened, and then the red is the predicted based on the equation with adjusting for the soybean meal diet as for adjusting the y-intercept. And you can see that it predicts very, very nicely for all of those, except when we get out to the tryptophan diet. The tryptophan pigs we did not get the benefit and growth rate that we expected when we increased the tryptophan level in the diet. But thought really the equation performed pretty nicely in that experiment. So I compared it then to some of the trials that University of Illinois ran and that Hans's group, and this was looking at increasing the leucine to lysine ratio with synthetic leucine, going from 100 all the way up to 300%. And again, I think the equation predicted the response there pretty nicely from those experiments as you move to those higher and higher levels. Hans's group also did this trial that looked at tryptophan to lysine by leucine to lysine interactions, and so did 3 tryptophan to lysine ratios within each of the 3 leucine to lysine ratios. And as you can see, as you go across here, they have some increase with the tryptophan addition to the diets, and really didn't see as much negative effect at the 200% leucine with using synthetic leucine as what we might have thought, but you really see that showing up here at the 300%. And when you look at the prediction versus what actually happened, again anchoring it off of the very first control diet, you see that predicted pretty well at the 23%, probably over-predicted at the 28%. Predicted the response in the 200% pretty nicely, but the actual, they did not reduce their performance as much as what the model suggested, but then again predicted pretty nicely on the 300%. So again, I think responding relatively good for what the model expectations were. Now I'm going to show you where it started going awry, and this is where we're trying to continue to learn what's going on in some of these trials where the pigs are not responding. So here's a trial that Hans's group did where we had positive control with soybean meal, replacing that soybean meal with 26% high-protein DDGs, and then adding either valine, isoleucine, tryptophan, valine and isoleucine, valine and tryptophan, isoleucine and tryptophan, or all 3 of the amino acids. And what we would have expected to have happen if it was just a branch chain response and the model would have been predicting it right, we'd expected some benefit to the valine, isoleucine, and tryptophan. We'd expect it to be a little better when we put especially the valine and tryptophan and together, and better with all 3. Well, that obviously didn't happen. And what they did get a response to this valine plus tryptophan, got some benefit, intermediate, I should say, between the negative control and the positive control. But I think it's one of the things we may be running into is some of this confounders that we have between different ingredients and different diets and some of the stuff that Dean Boyd talked about yesterday that we aren't taking into full account. We've seen some of the same thing with some of our trials. When we've tried to replace all the soybean meal in these diets with high-protein DDGs, the branched-chain amino acids don't always bring it back for us. If we maintain some level of DDGs, we've had much more success at bringing the performance back. But if you look at the other, there's a number of different things that were measured in this study that I think were really good. The one that I'm pointing out here is the plasma tryptophan and what happened there. And this is one that's intrigued me, and I'm hoping I'm not reading too much into this, but if you look at what happened in the negative control, how much the tryptophan got driven down, it's interesting to me how when some of these branched-chain amino acids are brought in, even though there's not significant differences here, the plasma tryptophan level went up quite a bit. And same over here, when you look at tryptophan and then adding valine with the tryptophan, you had even a higher, actual numeric higher level of plasma tryptophan than what the positive control had. We're starting to see this in some of these trials, that there's something still going on besides the blood-brain barrier interaction between these branched-chain amino acids and particularly tryptophan, and sparing effects that may be happening for tryptophan if we have high enough levels of some of those other branch chains. Showing you another experiment where we were unsuccessful with the model in terms of bringing performance back. This is a relatively recent trial that Bo Williams did with us that took, again, our positive control and then adding 5 to 7.5%, depending on the phase, of corn germ with 20% DDGs in all these diets. And you can see the loss in performance that we have with the negative control. And in this case, not only did we not see any benefit, but when we added the valine to this diet at a pretty high level, we actually saw a negative effect in average daily gain. We still don't 100% understand what's going on in this trial. And then Bo's most recent trial, and this guy said, we're going to take a step back and take a very practical approach of maybe we just need to make sure we have our ratios in a reasonable level. And these diets only had around 150% leucine to lysine, brought in by 30% DDGS early and then 20% in the last diet. And what you can see is that moving to the low ratio of amino acids, and so this has more soybean meal in it, replacing soybean meal with crystalline amino acids to drive the ratios down to what a lot of us were very close to formulating to in our diets, and then going to medium ratios or to high ratios for those amino acids. And again, the model would have predicted this to be a linear response here, and we do pick up that linear response, but we really don't see much benefit after going above that medium ratio of 60% isoleucine, 72 on valine, and 21% tryptophan. And then I want to head and step out and look at work that South Dakota State did. And Ryan Samuels, in the audience here, the group, I think David Kleiser did really nice studies working with Paul Klein. And this is just one of the studies looking at the valine to lysine response. And this is in the 40 to 70 kilogram pig. We got a really nice response up to around 70%, start to plateau, and 70% valine to lysine. Still, though, didn't bring performance all the way back to the corn-soy control in the experiment, but helped us understand that the response is probably over that 65% valine, certainly when we're talking about 30% DDGS diets. You can see the average daily feed intake response. The biggest feed intake response was going from that 60 to 65%, then a little smaller response all the way up to 75%. But where the 70% comes in again was the feed efficiency response of improving feed efficiency up to about a 70% valine to lysine ratio in those phases. But again, feed efficiency being a little higher than with the corn-soy diet. And then David did a series of experiments. I'm only going to show one other one of his, and that I thought was pretty interesting. Again, ties back to this whole soybean meal impact. And here he used the corn-soy diet, replaced all the soybean meal with high-protein DDGs, and had this a loss in performance, and then took that HP-DDG diet and either added soybean meal to increase the ratios for the amino acids, or he brought in synthetic amino acids to increase the ratio, or he blended the amino acids and the soybean meal to create the 50-50 treatment. And so you can see is he adding some soybean meal to that diet to bring the ratios up to that basal level, was able to bring the performance all the way back to the corn soy, but he was not able to do that with using just crystalline amino acids. And there's gobs of protein in this diet. On paper it should work, but again, there's something going on with that soybean meal that's given that extra performance in this particular study. And then the last study on branch chains that I want to show you is one that was recently published out of China that it took a little different approach, but the reason I'm showing it to you is because of some of the measures they made on fat metabolism in these pigs and what was going on with in the adipose tissue. So they have this is what we would more think of comparing a high protein to a low crude protein late finishing or mid finishing diet. They're from 59 to 95 kilograms. So you can see that they see this loss in average daily gain that many of us have measured when you go from a 16 to a 12% crude protein diet. You can argue that they were probably deficient in valine in this particular case. Then they took that diet and they added branched chain amino acids and they added the same amount of branched-chain amino acids, but in different ratios. And so they came up with a ratios of a 2:1 of leucine to isoleucine to valine, or a 2:2 of leucine, isoleucine to valine, or 2:1:2, where isoleucine was the lowest level, and they had the intermediate level of, I should say, of leucine, and then the highest level of valine. And so on the performance side, you could look at this as a cynic and say, well, all you have to do down here is look at 63, 69, we're still borderline, you get up to the high enough valine in the diet, and you bring them back to the control. And that might be all the response from growth performance is due to. But they did do some interesting measures to try to understand what's going on with this treatment, and in this treatment in particular, that they are performing poorer than, so why is it that valine outperform, in this case, performed better than the pigs that had excess isoleucine and what you'd think is pretty close to adequate amount of valine. And you look at feed efficiency, just what you'd expect with feed efficiency if you don't have enough either valine or enough amino acids in that diet for our crude protein level. You get this increase in feed efficiency. Here, though, you still have that poor feed efficiency, and particularly compared to when you're comparing these two diets on the end again. And again, the reason I thought this was really an interesting study is that they measured a number of different things in the adipose tissue as well as in the protein tissue to look at what was going on. And they found out that a lot of the gene regulation in the adipose tissue was changed by these branched-chain amino acid ratios. And so they found that both the low crude protein diet and that one that had the goofy isoleucine level, having too high of isoleucine relative to the valine content, had increased fat mass. They had increased serum fatty acid binding proteins and other fat markers. It upregulated expression of several of the markers for fat metabolism and reduced the expression of hormone-sensitive lipase. End up happening is those pigs got fatter, much like we see sometimes when we over-supplement synthetic amino acids replacing crude protein. But the most interesting part is that administration of those branched-chain amino acids in either the 2:1 of leucine, again, isoleucine, valine, or high levels of valine in presence of those higher levels of leucine, and ended up reversing much of those negative effects. And so if you get the ratios in the right aspect, it changed the fat metabolism to make it more normal. And so just my conclusions in terms of where I think we're at on branched-chain amino acids right now is I think first one is that realizing that it's difficult to separate out the impact of corn protein products when we're doing some of these trials from the branched-chain amino acids and the tryptophan effects. And that's why, of course, Hans did it with synthetic amino acids. But I think sometimes the synthetic amino acids also give us some things that aren't what's happening to us in the world when we deal with DDGS and corn gluten meal and so forth. Valine in the ratio between branched-chain amino acids impacts lipid metabolism. This not only has an impact that can grow finished pigs, there's been some interesting studies published out of China again recently about use of valine shortly before farrowing on increasing the colostrum fat content. And been several studies now that are showing the same kind of response. And it tells me that it is impacting fat metabolism more than what I was aware of that it certainly did. As corn protein decreases, and this is one other thing that we've certainly seen, is corn protein has been going down steadily in the United States over some time, and as corn protein levels go down, the ratio of these amino acids within corn protein also changes. The lysine stays relatively constant because of where it's at in that corn grain, but the branched-chain amino acids are the main ones that are reducing. I just showed the example here of looking at NRC's levels versus the Ajinomoto's most recent summary, and these are two different databases, so you have to be very careful here. But even if you use use some of the regression equations from the 1998 NRC or any of those, you can see that those branched-chain amino acids drop at a faster rate. And so in one case, you could argue that some of the negative effects that we're seeing become less and less of an issue if we lower our crude protein content in the corn grain. And so that's something you need to keep in the back of your mind. I think there's certainly we have evidence that particularly that when we have these higher leucine levels, that we should formulate at least near the upper end of the requirement estimates for isoleucine, and not argue that's somewhere around 60%. For valine, somewhere between 70 to 75%. You can argue it probably depends on your loadings on ingredients quite a bit and where that one would end up. And tryptophan, we can probably argue all day whether that should be 19.5 to 21%. But I think if you from what I know today, if you formulate within those windows, I don't think we have good enough evidence that says going higher than those levels is going to give us a payback in terms of protein deposition and growth rate in growing and finishing pigs. So in practical diets, what it really means is that valine really becomes more of an issue early in our corn-based diets with DDGS, and isoleucine is more likely to become limiting late, and of course tryptophan you got to watch all the way through. And so acknowledgements for our branched-chain amino acid work. I do want to give a lot of credit to Keith Hayden with CJ for being very important and involved with a lot of the studies that we've done. The Pork Checkoff for their funding, for both New Fashion Pork and New Horizon Farms for conducting a lot of the studies. I also wanted to make sure to give credit to University of Illinois and South Dakota State, and for Paul Klein and Christensen's for all the work that they've done on helping us get more and more data in this area. So now I'm going to switch gears and talk about training the next generation of graduate students. And I'm just going to cover really three main areas in just a couple of slides here. Is where are our students coming from? How are they being trained? And again, I'm going to speak a little bit about what our model is, and then finish up with just a little bit about the importance of maintaining swine programs at universities. And this has been a little bit talked about both yesterday and today, and so that is appropriate to end up on that note. First, in terms of where our graduate students have come from, I went back and looked at the last 60 graduate students that we've had in our program, and it didn't surprise me too much in terms of the first category based on the number of the students we've had through the group, but right at about almost 50% of our students have come from a show pig background. And so sometimes when I hear people and it's particularly some of our commercial industry, really hammering the show pig side, and I understand from a biosecurity issue that there are some struggles there, but the reality is a lot of our leadership in our industry and a lot of you in the room have grew up in that reality, and that's how you got and fell in love with pigs and wanted to move into the swine industry. And so we got to engage that group and interact with them as a potential pool of our future leaders. Internationally, we're just right about 25% of our students have been international graduate students. Most of them come in with a base veterinary training, which is, of course, most of you would realize that it's a little bit of a combination of like our animal science undergraduate with a vet med, not quite as much vet science as what our veterinarians would typically go through, but a mixture of those two. So really, really good base training. We need to obviously encourage and have a pipeline to bring those students into our industry. Then we have about just under 20% of our students that came from where their family was in production or themselves worked in production before coming back to grad school. Again, an important pool. And then a growing group, more in the recent years than what the long further past, and ties into the importance of maintaining swine programs at other universities in particular, is pre-vet, pre-med, or pre-dental students that end up deciding that that's not what they want to do with their life and change direction and go into swine nutrition. And that's an area, I always use Chad Polk as one of my best examples. He's a pre-dental student that got into Michael Zane's swine production class at University of Georgia and fell in love with the whole pig concept and pig production, and decided that's what he was going to do for his career. And so there's a good example of Michael Zane was not at University of Georgia. When he retires, if they don't refill his position with somebody that's passionate about showing students, we may not have a Chad Polk in our industry, we may not have a Paul Klein in our industry that also went through the University of Georgia. So some of those things that why it's important to think about some of our universities on the periphery that you may not think of as major swine states, but I know we have several people in the crowd that also have come through places like that and end up in the swine industry. But similar to like many of you and many of myself is when we were coming through school and we were young, we had some of these experiences. I had certainly a little bit of that production background. I had absolutely zero clue about anything international. I was a country kid from North Dakota, but I ended up with this importance of maintaining swine programs. Joe Crenshaw is the reason that I'm in the swine industry, and I need to give Joe credit because he taught swine production at North Dakota State University when I was there, and so when I was looking at grad school, I was thinking, well, I was either going to ruminant nutrition and that was probably where I was going to go because I grew up mostly cattle and pigs, and Joe said, well, you should think about going into swine, and here's a few people you should call, and so that's how I got into the swine industry. And I really want to thank all of the groups that offer internships because, as I said, providing that opportunities to students. I asked our administrative assistant to just go out and find me the 20 Swine student internships, the first 20 you can find, grab the logos from those companies and put them on a slide for me. And these are the ones that we got up there. And I know we have a lot of other organizations that provide internships, and it's really important to get the training for our students as an undergrad, the ones that come in, don't have any production background, getting their feet wet, understanding what's going on in pork production. And so I do want to thank all of you that work for companies that provide membership, internship opportunities for undergraduates. For any of you that provide internship opportunities, would you please stand up? I know there's a lot of you in here. Thank you. Please give them a hand. It's a very, very important role for training undergraduate students and getting them exposed to the industry. And then all of you that have allowed graduate, undergraduate, or veterinary students to conduct research in your facilities. Any of you please stand up? No, we got several of them. Yeah, there's another group. Thanks to all of you. Very nice job. We really appreciate that. Any of you that have funded research projects where graduate students have been involved, and I know there's a big group of you in here. Yep, that's excellent. Thank you very much. Appreciate your help. All right, Bob, here's your chance to stand up. All of you that work directly with training graduate students at universities, please stand up. Pedro, Joe, everybody that's, yep, thank you. Thanks you for everything that you're doing. All right, now for all the students that are in the audience. All right, let's see how all the students. All right, I want you to I hope you looked around while everybody else was up and take the opportunity to thank those people as we finish up today, because it's because of all the investment that they're putting into your lives that you're going to be able to have a place in this industry that we all think is pretty important. So thank you for all the hard work you're doing and helping us. Okay. So how do we go about on the training side? Well, one of the things we do is this undergraduate internships. If they don't come to us with undergraduate internships already, they want to come to grad school, we really, I'd say it's probably been pretty firm that they have to do an internship before they get in our program, because they need to get some production understanding before we take them on. And then we have fellowships with several of you I know in the audience, and several that aren't here, where the fellowships are a very important part for training our students, where they not only managing research pigs, but a lot of times it's managing data, communicating remotely, learning how to deal with the timeliness and the demands of the real world and what production wants. And I know sometimes we don't turn it around as fast as what some people in production really want us to get it back to them. But the reality of helping them learn how to communicate, how to operate remotely, how to manage pigs that they never see, because that's what their life becomes a lot of cases. So helping them with that kind of training. Then the base research training is understanding the economics and understanding the end use of their projects. And I know that obviously that's a very important piece, is where that data is going to be used in the future. And this is an area that I know there's a lot of discrepancy between universities and a lot of different thoughts on how this one should be handled. We tend to handle our students where they manage multiple types of projects during their program from a wide, diverse area. And you can argue whether that's the right way or the wrong way. That's the way we do it, because we feel like where we're training our graduate students for, they need to be able to handle a wide breadth of things that come their way and be able to handle it relatively quickly once they get on the job. One of the ways we help them prepare for that is our prelim exams. We've totally changed our process there. We used to do the written exams, years ago, the way that I took written exams going through my PhD program. We don't do that anymore in our group. It's still an option if a student chooses to take it. They never choose to take it in our group, but we do. They can be options of grants, literature reviews, meta-analysis, decision tools. A number of the tools that you'll see on our website have been developed through some of those prelim exams, and gives them a lot of tools that I think they use throughout the rest of their careers. And I know that graduates, obviously for many of our programs, still have an awful lot to learn. And they know, the first thing you hear is how much they learn the first 6 months, first 9 months out on the job. But obviously our goal is to understand we have very high quality people, and we hope to train them with a team mentality that they're going to work together with others when they leave our place. And now I'll finish up with just maintaining swine programs at universities. As all of you, when you have the opportunity, encourage the university presidents, deans, department heads in your state or wherever you graduated from to maintain faculty of swine interests. This is really important in states like Iowa, obviously, but it's also important in states with small pig populations. I already talked about, used Mike Zane as an example at University of Georgia. You could use numerous examples across the country, not only because of you might find those diamonds in the rough of those individuals that become a more important part of our industry, but it's also, think about the number of students that have gone through the swine production courses that never have anything to do with swine, but they've learned a little bit about the background and a little bit about the swine industry and have more knowledge than what the average citizen has. When you make trips to campus, I know a lot of people we really appreciate help mentor our students, recruiting undergraduates, and also take the time when you're there to discuss the needs with administrators. Help them to understand how important having swine programs are. And then, this was already talked about yesterday, and this was not planned at all with Trey in terms of the endowment side. This is one. None of us thought that we would have to be looking for funding to replace ourselves at universities. But the reality is that if you're going to maintain swine faculty positions at universities, we're going to have to figure out a way to pay for it ourselves. It really hit me. I expected that might be the case for us in Kansas. Kansas, we're the number 10 swine producer in the country. If we have a beef position come open, it's filled like that. We have a swine position come open, they start going, well, what can we fill instead of the swine position? And so we kind of beg, borrow, and steal over the years to build the group that we have. But when I heard yesterday that's also the case here in Iowa, the number 1 pig producing state in the country, that shows you the current state of how university administrators and how they're making decisions about positions. It's when the budget is tight, they don't care what the position is. It's killed. So we need to control our own destiny in that area. And that ties into this, will the U.S. continue to be the center for swine research? When I was starting out, U.S. was just taking over that role from the U.K. in swine nutrition world. The U.K. was really the center of swine nutrition expertise when I was getting into graduate school and was really transitioning to the U.S. at that time. I think we're on that precipice right now that we're going to transition to China very quickly if we don't be careful. And the effort that Hans is doing is tremendous with what you're doing with the net and the chambers and stuff. We need to continue at all of our universities to try to maintain programs to be able to keep us towards the area of the forefront. I got 2 slides to finish up. This is the last 3 groups of graduate students through our program. I do think the future is very bright. You can be skeptical and pessimistic about all the education and what's going on in universities, but when we have the opportunity to work with the quality of people that we have, it makes my job worthwhile and fun and wanting to come to work every day. And so you look at, I think we probably have about 70% of the people in these 3 slides are in the audience today, and so that's also really a joy to see. And then I want to finish up with thanking John for the leadership in the swine industry and your mentorship to so many outstanding young people, not only the graduate students that we had here talked about yesterday and all the graduate students that you've personally touched here at Iowa State, but obviously a number of the faculty here and a number of us faculty across the U.S. that you've helped over the years. So really want to thank you for that. With that, I will turn it over to you, Spencer.
Thank you, Dr. Tokach. We'll now open the floor for questions.
Mike, thanks for a great presentation. It seemed like in your data you showed some effects that maybe those younger pigs were harder to overcome that excess leucine. We ran a trial at one of our research facilities. It seemed like early on, pre-120, We could not overcome that with supplementing the branch chains. Post 120, it was much easier. Is it something about that mTOR in that younger pig that might be contributing to that?
I'm not 100% sure. It's a great question. I'm not 100% sure. And then when we did the modeling, I mean, Enrique had tried to model from weaning to market initially, and the nursery pigs truly did respond differently, and we could not explain the performance in nursery pigs with the model, and so we chose to model only finishing pigs in that. And so there's something else going on there in the young pig that we don't fully understand that with. But I think some of it is you want to be careful on where exactly how high that leucine level gets, because I really do think that, especially in younger pigs, if we start getting that leucine level beyond 165, 170%, I think we just can't ever bring them back. If you're dealing in the 130 to 160, you probably do get some benefit to increasing the branch chains.
Mike, just two separate questions, I guess. The first question is a lot of your work is using corn DDGs, corn proteins, right? We do know there's variability about, digestibility of lysine, tryptophan, so on and so forth. So how do we interpret that data, to a broader audience, I guess?
That's a good question. I mean, one of the things we did, of course, in those trials, we analyzed all the ingredients ahead of time and tried to account for at least the variation in the amino acids that were in the product. But we did use standard digestibility values, and so the differences in digestibility we would not have taken into account.
The separate question is, the isoleucine, right? In the young baby pigs, isoleucine to lysine ratio. I think right now they're in Europe, in Denmark, they just updated their requirement or recommendation. They actually adjusted it down to 50. I was thinking maybe that's because they're using a lower protein, diet. Do you have any comments on that?
Yeah, I mean, we've done those isoleucine trials, and honestly, if you get the leucine level down to that 100 to 110%, the argument is whether whether you get much benefit over 48%. I mean, we've Annie Lerner is in the audience here. She did that with I think 52% was what her trial showed. And so in that young pig, if you don't have excess leucine, I do think you can get that isoleucine level down that low. You just have to. Anytime you start pushing those lower ends, though, the point you brought up earlier is you better know your ingredients pretty darn well, because you start pushing that bottom end. If you do anything wrong, we know with branched chain amino acids, it falls off a cliff pretty quickly in terms of feed intake if we're below the requirement. Yeah.
Hey, Mike. First of all, thanks for letting me be a part of your program so many years ago. Thinking about the work that's been done in Canada and elsewhere in small grains diets with wheat and barley and their tryptophan to lysine recommendations, how do you square that with the work that you're doing with the corn byproducts? Because obviously different fiber sources, as you showed here, different ratios to the other large neutrals. How does that all sort out? Because the recommendation is 20, 22%, still about the same.
Yep. Yeah, and I think the the issues with the tryptophan, especially, you also have to look at some of the what are the other amino acids and where the crude protein is in those diets, and we did again, if you go back to some of Annie's trials, we did some trials where if you were at the pig's requirement for amino acids, what the response was to amino acid ratios, and if you're below the requirement, what the response was to the ratios, and we titrated the lysine requirement first, and with the titrated requirement, you get by with lower ratios, you get the same level of performance, but when you went below their requirement, and this is particularly in Europe, as there's pressure on crude protein, they've ended up going below their requirement earlier than what we've done, and in those, especially the earlier nursery diets, and when you go below the requirement, you do want to push towards the upper end of any of those estimates, and so that's where that 21% tryptophan response, and we've seen the same thing in our trials. If we go below the requirement enough on lysine, the response to the other amino acids increases, and so I think that's part of what the differences are there, Joe. Yeah.
I think we might have time for one more question, if anybody has one.
All right.
All right, Please join me in thanking Dr. Tokach.
Thank you.